everyone, my name is Gurbaxkor from Mount Sinai Hospital, and I'll be presenting the Phase 2 registrational study of Anitocabtagene Autoleucel for relapsed and or refractory multiple myeloma, the updated results from iMMagine-1. These are my disclosures. Anito-cel is a CAR T-cell therapy that targets BCMA and employs a CD3 zeta signaling domain and a 4-1BB co-stimulatory domain. Unlike other CAR T-cell constructs, the binding domain is a novel synthetic protein called the D domain. D domains are triple alpha helical structures that were originally designed for simplicity and stability and later repurposed as antigen binders for therapeutic applications. Due to its small size and architecture, the D domain construct used in anito-cel results in high protein expression, leading to high transduction efficiency. That is, percentage of cells that are CAR positive and high CAR surface density. As a result, anito-cel's dose comes with a low number of CAR negative cells and a low total T cell dose. Because of the simple structure, D domain CARs are stable and remain highly expressed, but lack tonic signaling due to rapid folding, lack of disulfide bonds, and stable hydrophobic core. Additionally, anito-cel's D domain binder has a fast off rate, meaning that it is able to quickly release from the BCMA target. The combination of high D domain CAR surface expression and the D domain's binder's fast off rate may allow for optimal tumor cell killing without prolonged inflammation. By way of background, anito-cel was originally evaluated in the first-in-human dose escalation phase 1 trial in patients with relapsed or refractory multiple myeloma in the fourth-line plus setting. As presented at ASH 2024 at a median follow-up of 38.1 months. The data from the phase 1 demonstrated a complete response or better in 79% of the patients with a median progression-free survival of 30.2 months in the full population and 34.3 months in patients with a CR or better. The median overall survival is not reached. The safety profile is predictable and manageable with no delayed or non-ICONS neurotoxicities, including no Parkinsonism, no cranial nerve palsies, and no Guillain-Barré. iMMagine-1 is the pivotal phase 2 trial evaluating anito-cel in patients with relapsed and or refractory multiple myeloma. Eligible patients were required to have had prior exposure to an IMiD, a proteasome inhibitor, and an anti-CD38 monoclonal antibody, to have at least received 3 prior lines of therapy, and to be refractory to the last line of therapy. Optional bridging therapy was permitted at the investigator's discretion. The primary endpoint was overall response rate per 2016 iMWG criteria. The data presented here is investigator assessed response. 129 patients were enrolled and leukapheresis. 11 patients discontinued prior to lymphodepletion, and 1 patient discontinued between LD and anito-cel infusion. 117 patients received anito-cel infusion and comprise the full study population for iMMagine-1. The safety and efficacy data presented today are based on a data cutoff of May 1st, 2025, with all patients having at least 4 months of follow-up with post-anito-cel infusion. Anito-cel was successfully manufactured for 99% of the patients enrolled in iMMagine-1. The patients enrolled in iMMagine-1 are representative of fourth-line plus relapse and refractory multiple myeloma population. The median age was 64 years, 50% were 65 years or older, and 9% were 75 years or older. 76% are white, 15% are Black or African American, and 9% are Asian or other race. 49% of the patients have had at least one high-risk prognostic feature, including bone marrow plasma cells more than 60%, ISS stage 3, extramedullary disease or high-risk cytogenetics. 100% were refractory to the last line of therapy, with 86% being triple-class refractory and 40% being penta-drug refractory. The median prior lines of therapy was 3, with 51% having 3 prior lines of therapy. 9% of the patients received anito-cel infusion in the outpatient setting. With a median follow-up of 12.6 and minimum of 4 months follow-up for all patients, the overall response rate is 97%, with a CR or better rate of 68% and a VGPR or better rate of 85%. as seen in other myeloma clinical trials with longer follow-up responses, have deepened, with the complete response or better increasing to 68% compared to 62% reported at ASH 2024. It is anticipated that depth of response will continue to evolve with more follow-up. 75 patients were evaluable for MRD at a sensitivity of 10 to the minus 5, and 93% or 70 out of those 75 were MRD negative at this level. Both the median time to first response and median time to MRD was 1 month. The estimated 6-month PFS is 91.9%, and 12-month PFS is 79.3%. The estimated 6-month OS is 96.6%, and the 12-month OS is 95.2%. With many patients in ongoing follow-up, it is likely that these estimates will continue to evolve with further follow-up. In terms of toxicity, 85% of the patients had cytokine release syndrome of grade 1 or less, including 15% of the patients with no cytokine release syndrome. Of the 85% patients who experienced CRS, the median onset was 4 days and a median duration of 2 days. 80% of the patients had either no CRS or had resolution of CRS by day 7 post-infusion of anito-cel. Similarly, 97% had either no CRS or had resolution of CRS by 10 days post-infusion. For those patients experiencing CRS, management was in line with standard medical practice, and no prophylactic tocilizumab or dexamethasone was utilized in any patient. 71% of the patients had either received no dexamethasone or a single 10 milligram dose of dexamethasone for CRS management. One patient did experience a grade 5 CRS event. To provide context, this was a 76-year-old patient who had disease that was rapidly progressive between screening and baseline and who did not respond to bridging therapy. This outcome highlights the importance of bridging therapy in the overall CAR T-cell treatment journey to ensure optimal safety and efficacy. In terms of ICONS, 8% of the patients had ICONS of any grade. All cases resolved. There was no delayed or non-ICONS neurotoxicity were observed, including no Parkinsonism, no cranial nerve palsies, and no Guillain-Barré at a median follow-up of 12.6 months. Similarly, no delayed or non-ICONS toxicities were observed in the phase 1 study with a median follow-up of 38.1 months. The median time to ICONS was 7 days. The maximum time to ICONS was 10 days, except for one patient who experienced grade 1 ICONS with confusion on day 31 that was rapidly resolving. The median time to ICONS resolution was 4 days. The maximum duration of ICONS was 12 days, except for one patient with grade 2 ICONS who had time to resolution of 29 days. Collectively, ICONS was uncommon, typically low grade, and resolved in all patients. Most notably, there was no delayed or non-ICONS neurotoxicities. In terms of the most common grade 3 or higher treatment emergent adverse events were neutropenia, anemia, and thrombocytopenia. Non-hematologic grade 3 or higher treatment emergent events were uncommon, with grade 3 or higher infections being only 9%. No cases of immune effector cell-associated enterocolitis have been reported. No replication-competent lentivirus was detected, and no secondary primary malignancies of T-cell origin or hematologic malignancies were reported. 3 deaths occurred due to treatment emergent adverse events: a retroperitoneal hemorrhage secondary to a biopsy in the setting of a plasma cell leukemia, which developed between screening and baseline with grade 4 HLH at the time of death. This was a complex case, and notably the only case of HLH to date, cytokine release syndrome, and a fungal infection. In conclusion, anito-cel is a novel BCMA-directed CAR T-cell that uses a novel synthetic compact D-domain binder, which facilitates high transduction efficiency, CAR positivity, and CAR density on T-cell surface expression. It has a fast off rate, which allows for a predictable and manageable safety profile. The overall response rate was 97% with a CR or better rate of 68% per iMMagine-1 criteria. 93% of the MRD evaluable patients were MRD negative at 10 to the minus 5 or lower. The median PFS and OS was not reached. The 12-month PFS was 79%, and OS was 95%. 85% of the patients did not have CRS or had max grade 1 CRS. 92% of the patients did not have icons. And once again, there was no delayed or non-icons neurotoxicities, including no Parkinsonism, no cranial nerve palsies, no Guillain-Barré syndrome, and no immune effector cell-associated enterocolitis. More than 150 patients have been dosed across the phase 1 and phase 2 anito-cel programs. Anito-cel demonstrates deep, durable responses in fourth-line plus relapsed refractory multiple myeloma with a manageable safety profile. And that is it. And this is iMMagine-3 clinical trial design, global phase 3 study that is now enrolling. It is being co-developed by Arcellx and Kite. It is evaluating patients in 1 to 3 prior lines of therapy. That's it. Thank you so much. We'd like to thank our patients, our families, and the caregivers, the staff, and the research coordinators and investigators at each participating institutions.